Animal viruses, part 2 audio. So the first part was just an introduction as to why our understanding of human and animal virus replication biology lag behind our understanding of bacterial viruses. So this second audio, you guys, it'll be like the first 1 or 2 steps in animal or human virus replication. And what I'll try to do here, folks, is try to use the pandemic coronavirus and then another virus of great concern to us, influenza virus, for some of our specific examples. Okay, so this first slide is the very complicated replication cycle of coronavirus. So you won't have to learn it in the detail that's presented here, you guys. So we're going to go through it just step by step. So just so you know, this is the our coronavirus, and this would be the surface of the in our case, you know, if this is a human coronavirus, this would be the surface of a human cell. Okay, but remember, folks, a lot of the coronaviruses, they're zoonotic. So this could also be the cell membrane, cytoplasmic membrane of a non-human animal host. Okay, and then this is a little bit confusing here, you guys. So this is the endoplasmic reticulum. It's not the nucleus here. Endoplasmic reticulum, just to keep you oriented. Okay. So, folks, we will be talking about how the viruses have to attach to their host cell. So we want to know what the adhesins are, and so this is just a model of the coronavirus. It is, and again, I should have written this in here. So the coronavirus, you guys, it's an enveloped RNA virus. It has a single strand of what's called positive sense RNA, and we'll get back to that in a little bit, what the heck we mean by positive sense RNA. So again, folks, this represents the stolen host cell membrane, and we'll see with the coronaviruses, they steal membrane from a structure inside our cells called the endoplasmic reticulum. Okay, and then before they steal the membrane, before they're released from their cells, envelope viruses have to insert viral proteins, glycoproteins, into the stolen cell membrane because remember, at the very least, the virus has to get its protein adhesins into that stolen host cell membrane, right? So this little lollipop-looking protein structure here, this is the coronavirus S protein. Okay, looks like a little spike here, and that is an adhesin, and we're going to see in the novel coronavirus, the pandemic strain now, and in the previous 2003 SARS coronavirus, these adhesins are going to bind to a specific surface receptor on cells in our lungs called the angiotensin converting enzyme receptors, the ACE receptor. So we'll come back to that. And then, folks, again, this is not, I don't want you to worry too much about it. This HE protein, hemagglutinin esterase protein, this might be a co-receptor, a receptor that helps the coronavirus bind more strongly to our cells. And to me, this is fascinating because they think this co-receptor might bind to sialic acid receptors on the surface of our cells in the lungs. And the reason to me that's fascinating is that's exactly the receptor that influenza viruses use to attach to our cells in our lungs. They use a receptor that binds to sialic acid receptors on the surface of our lungs. So to me, you guys, it's just fascinating seeing the similarities and differences between coronaviruses and our influenza viruses. So let me just say this, sorry, you guys. So we're going along. Okay, and then I think, folks, the next model here is I want to show you influenza virus. So these are the two viruses that I'll try to be kind of using as examples and comparing and contrasting. So, folks, the influenza virus, there's three groups, three types, type A, B, and C. The ones we're really worried about are the type A influenza viruses. These are the ones that are zoonotic and have the potential to wipe out millions. It was a type A virus that caused the 1918-1919 influenza pandemic. It was a type A that caused the 2009 influenza pandemic. Okay, so the influenza virus, it too, and again, I should have included this in the text. It's an envelope virus, so it's going to steal the cytoplasmic membrane of the cells in which it's replicating. And again, folks, before it steals our cytoplasmic membrane, it has to insert its viral proteins into the membrane, into the envelope. So here, folks, it's kind of hard to see these yellow spikes. That's the adhesin of influenza virus, and it's called hemagglutinin. It's sometimes called the H antigen. And the hemagglutinin is going to bind to sialic acid receptors on the surface of our cells in our lungs, right? So that sialic acid is pretty fascinating. We're going to see, you guys, if we can make antibodies against the H antigen, it will block the virus from attaching to our cells, and blocking attachment is called neutralization. So neutralizing antibodies against the H antigen are going to be protective, right? So if we survive an influenza infection, we'll have neutralizing antibodies against that specific strain. If we vaccinate, um, with a specific strain of influenza, we'll have neutralizing antibodies against that specific strain of influenza. Another envelope protein, you guys, that's going to be really important is going to be this guy called neuraminidase. And we're going to see the neuraminidase, sometimes called the N antigen, plays an important part in how the viruses escape from our cells. So we'll come back to that. Now, influenza virus, I better type this in, you guys. I apologize. Again, it's going to be enveloped. It's enveloped, and it's what's called a negative sense RNA virus. And very importantly, you guys, it has a segmented genome, meaning it has 8 RNA segments, like little, almost like little mini chromosomes. And each segment carries different genes. And we're going to see that's going to be really important in permitting influenza viruses to undergo a process called reassortment, where 2 different influenza viruses can infect the same cell, and then the RNA segments can mix. So then when the newly replicated influenza viruses are released, we'll have brand new combinations of little RNA segments in there. And as a result, you guys, we can get, in the newly replicated viruses, we can get brand new combinations of this hemagglutinin and neuraminidase. And we think that that phenomenon called antigenic shift might be responsible for the worldwide epidemics, the pandemics that we see every 20 years or so. Okay, so we better save that. Okay. All right, so just to give you an idea of the receptors for the coronavirus, excuse me, not the receptors, you guys, apologies, the adhesins of the coronavirus and the influenza virus. Okay, so remember, folks, the first step in viral invasion of our cells is they have to be able to attach to our cells. So step 1 is attachment. Remember, the fancy name for viruses attaching is called adsorption. So for the viruses to be able to attach to our cells, they have to have surface adhesins, right, that bind to specific host cell surface receptors, complementary molecules on the surface of our cells. So recall, folks, we said for the influenza virus, the adhesin is called hemagglutinin, that H antigen, and it's going to bind to the host cell sialic acid receptors in our lungs. Okay. And then for this novel coronavirus, and you guys, this is the fancy scientific name for this new pandemic strain. So the first SARS, severe acute respiratory syndrome virus, caused a pandemic in 2003. So this one, you guys, I know this is going to be confusing, but this is like a cousin, a close cousin of that SARS, 2003 SARS. So they're calling this guy, this novel 2019-2020 coronavirus, SARS-CoV for coronavirus -2, right? But in the news, and everybody's talking about COVID. So this is the disease that this pandemic coronavirus causes. Okay. So what's fascinating, you guys, for both the 2003 SARS and for this 2019-2020 pandemic coronavirus, remember those S protein spikes in the envelope? It's believed, or at least in the report that I linked here, you guys, or excuse me, on the next slide, I'll give you the link to the report, the study from which I got this information. Oh, and also I got it from a very good medical microbiology book. The S protein of both of those human coronaviruses in our lungs bind to what are called the ACE2 receptors. And again, you guys, I think, and A&P folks yell at me if I do this wrong, this is angiotensin converting enzyme 2 receptors in the lung. And A&P folks, you guys could school me on this, but this is that angiotensin is part of that incredible renin angiotensin aldosterone system that's just amazing, you guys. Maybe maybe I can give a reference to that system. And those of you that have had A&P, I'm sure you've studied it. It's just amazing. Okay, so the coronavirus S protein is going to bind to these ACE2 receptors in our lungs. And then remember, you guys, we think that hemagglutinin esterase protein might be a co-receptor, and it might be binding to that sialic acid receptor. So this is the fancy name for this specific type of sialic acid and acetylneurominic acid, and we'll just call it a sialic acid receptor. Okay, and this is just a Wikipedia link that tells you about an acetylneurominic acid is a type of sialic acid. So you guys, this was the study that just to me blew me, oh my God, just blew me away. Okay, so this is the link from which I'm getting this information, right? Because you guys know I'm not a coronavirus expert by any means. Okay, so in this study, right? In this study, you guys, I've got it right here. This was published, I guess, in March 23, 2020, or maybe that was a date I accessed it. Apologies. And this is by a couple, James Diaz, MD, MPH, Doctor of Public Health. He's a professor and head of environmental health sciences at Louisiana State University School of Public Health. And then, let me see here. There was another co-author, and I guess I've lost. I thought there was another co-author. Okay, apologies, there's not. But the title of his paper is ACE inhibitors and angiotensin receptor blockers may increase the risk of severe COVID-19. Okay, so this was his hypothesis, is that, okay, so since the 2003 SARS and now our new 2019, 2020 coronavirus, they both bind the angiotensin converting enzyme 2 receptors in the lower respiratory tract. Okay, so we think that's their target. That's why we're getting these. And a lot of folks are developing really serious, potentially fatal lower respiratory tract pneumonias by the virus. They talk about taking radiographs and the lungs having this ground glass appearance, which I guess is classic for the viral pneumonia. So Dr. Diaz's hypothesis was, will the use of ACE inhibitors, so angiotensin converting enzyme inhibitors, and angiotensin receptor blockers cause an increased expression of coronavirus receptors, thus increase viral attachment? So you guys, I've had a very interesting personal experience with this. So this last week, my blood pressure skyrocketed. It was like scary. It was so high. So I had just read this paper. So when I went into the doctor, I'm like, I know that very often people with high blood pressure, they'll put you on these ACE inhibitors or these angiotensin receptor blockers. And I talked with the doctor. And I said, yeah, I just read that, if I start taking those, it might increase my risk for serious coronavirus infection. And she was like, yeah, I heard about that too. So they put me on a diuretic instead, and it seems to be working. But you guys, remember in the news, we're hearing about folks that are older. They seem to be at much higher risk for dying from the pandemic coronavirus. So, one hypothesis that Dr. Diaz was proposing is a lot of us old folks, what are we on? We're on blood pressure medication, right? And the next step, I guess, for me is going to be one of these, right? These ACE inhibitors or angiotensin receptor blockers. So, what if, in addition to being older and our immune system doesn't work so well, if we're on these drugs to decrease blood pressure, could that also be increasing viral attachment, coronavirus attachment, right? Because in this paper, it was discovered that people given these drugs, they had an increase number of the coronavirus receptors, right? So this is fascinating, guys. And certainly, like my doctor said, she goes, no one should ever stop taking these blood pressure medications because, the high blood pressure can kill you or cause serious harm, right? And that's more likely to kill you than the coronavirus is, right? But this is something that maybe finally, when the pandemic is over, more studies can be done on these ACE inhibitors and angiotensin receptor blockers. Okay, so again, folks, here's the link to the study. And again, don't nobody if you have parents or grandparents on those blood pressure medications, don't tell them to stop, right? This is just kind of nerdy, fascinating to me. Okay, so then, folks, we'll just finish up with this. So step one in virus replication, the viruses have to attach to our cells. So here's my goofy cartoon. Here's a host cell. Maybe this is an epithelial cell in our lungs, and these are the host cell receptors right here. Okay, and this is kind of generally talking about um, microbes and toxins attaching to our cells. So let's focus on this guy right here, you guys. Let's pretend this is either the pandemic coronavirus or it's influenza virus. And these little protein spikes coming out here, those are the adhesins, right? So for the virus to infect our lung cells, those adhesins have to bind to complementary host cell surface receptors. Well, a way we have evolved, animals have evolved a way to, vertebrates rather, have evolved a way to protect ourselves against viruses attaching to our cells or bacteria attaching to our cells or toxins attaching to our cells. And the way we protect ourselves is we make these cool proteins called antibodies or immunoglobulins. And you can see here, this is a cartoon for the predominant antibody in humans called IgG. It's a Y-shaped molecule. And right here at the tips of what look like arms, you can imagine there's little hands. And those hands can bind to complementary molecules. So the hands that are going to bind to these complementary molecules, the hands are called antigen binding sites. And the molecules to which they bind are called the antigens. Antigens are substances which will trigger a specific acquired immune response. So how is this helpful? Well, if the antibodies bind to the adhesins, can the adhesins bind to the surface receptors? And the answer is no. So the antibodies are blocking attachment, and it has a fancy name. It's called neutralization. So when you block attachment of a virus or a bacterium or a toxin, we call it neutralization. And thus we're going to call these antibodies, what kind of antibodies? These are neutralizing antibodies. And neutralizing antibodies, again, you guys, they can protect us from serious infection and harm by viruses, bacteria, and even bacterial toxins. So again, this is how we have evolved to protect ourselves against viruses. So, for example, you guys, let's say I get the pandemic coronavirus infection. Hopefully, I will survive, right? And then what at least for a while, my body will have, as a result of the infection, my body will have made these neutralizing antibodies. And hopefully these neutralizing antibodies would be circulating in my body for at least a year or so, right? So hopefully my chances of getting reinfected with the same strain of coronavirus would be greatly reduced. Well, the problem is, right, a lot of people aren't surviving their first infection, right? So um, that's where the need for a vaccine is absolutely crucial, you guys, because that's the goal of many of our vaccines. We vaccinate our kids ourselves, and the vaccines trigger our bodies to make these neutralizing antibodies before we get infected. And that way, if after being vaccinated, if we encounter the real lethal coronavirus, right, we already have these neutralizing antibodies circulating, and they'll bind to the coronavirus and prevent it from infecting our cells. It can't replicate, and voila, right? We won't suffer serious infection, and hopefully we won't die, right? Sometimes vaccines can't prevent 100% viruses from replicating, but they'll definitely decrease the number of viruses that can attach. They're going to decrease viral replication, and thus, at the very least, we hope these vaccinations will protect us from serious harm or death. So we really need a coronavirus vaccine, you guys. So folks, what we'll do is we'll stop here. So we'll say this is animal virus part 2, and then on the next video, you guys, we'll see what happens after the virus attaches to our cell. So attachment, adsorption is step 1. Step 2 is virus entry. So this is where we'll start. We'll see there's 3 different ways animal viruses can get into our cells. Okay.